Welcome back, class. So, we are talking about how plants get the materials they need. And then, once they have the materials they need, how they transfer and transport across their entire plant cell body. So, we are going to look at plant cells as having two different compartments. One is what's inside the cell membrane, and one is what is outside the cell membrane. So the apoplast is going to be everything exterior to the cell membrane. This would be the space that the cell wall takes up, extracellular spaces, and also dead cells, the interior of any dead cells, such as vessel elements or the tracheids of vascular tissue. So if water and nutrients are going through the apoplastic route, they're not going into cell membranes. They're just going from cell wall, cell spaces. Just that is the apoplastic route. The symplast is going to be what's in the cytosol of living plants. And if you remember, that cytosol is connected by plasmodesmata. So the symplastic route is going to be get into the first cell, and then once you're in the first cell, you just go from cell to cell through those plasmodesmata. That's the symplastic route. There's also the transmembrane route, which is a combination of both. You come into a cell, you go through the cell membrane, but then you leave the cell through the cell wall, then back into the cell, then back out of the cell, then back in. So you're going across membranes again and again, which is why it's called transmembrane. One of the things that is going to be happening is how are we moving solutes in and out of cells. So the plasma cell, the plasma membrane, has the ability to control short distance movement of substances. Some things can go right through the membrane. Some things are going to go through transport proteins. There are various transport proteins that help make solutes move across the cell membrane and help them enter either the symplast, the apoplast, or the transmembrane route. Water can move right across a cell membrane. When water is absorbed or lost by a cell, that's called osmosis. And water potential predicts what direction water will move. So here is a bunch of words. What is easiest to me is to remember water follows salt. So if there is pure water outside of the cell and more salt and sugars, more stuff inside of the cell, water moves into the cell. If there is more sugar and salt and all kinds of solutes outside of the cell, the water will move out of the cell. So in a hypertonic environment where there's lots of solutes outside, the water moves out, the cell shrinks. Hypotonic environment, not a lot of solutes outside the cell, so there's more inside, water comes in. That's hypotonic. In a hypotonic environment, you have a nice turgid plant. In a hypertonic environment, you have a wilted plant, eventually a dead plant. So solute potential is the same as osmotic potential or solute concentration, and that determines which direction water moves. Pressure potential is the physical pressure on a solution, so it's positive or negative in relation to the atmospheric pressure. Water will move from positive pressure to negative pressure. This isn't seeing solute concentration, this is just the actual pressure, physical pressure being applied to a solution. Water in living tissues is usually going to be under positive pressure, and in dead hollow tissues under negative pressure. So water will move from living tissues to non-living spaces. So that all determines which direction water is going to move. There are also aquaporins, which are channels in the cell membrane that allow increased movement of water. So water can move through these aquaporins. Aquaporins will allow osmosis to happen. They can also close or open as a response to what's going on in the cytosol. So if there's more calcium in the cytosol, those aquaporins can close. If the pH decreases, if they're in a more acidic environment, those aquaporins can open. However, diffusion, although it's great on an individual cell basis, it doesn't go over long distance very well. It would take like centuries for that water to get up to the top of a tree. By then, your tree is dead. So there's something called bulk flow. We're not waiting on diffusion. We're going on bulk flow to let water move over long distances. Bulk flow will be regulated by pressure gradients. So fluid will be flowing from high pressure to low pressure, independent of concentration. And bulk flow is going to occur in xylem and phloem of vascular tissues, in tracheids and vessel elements of the xylem. The cytoplasm of sieve tube elements in the phloem is almost devoid of organelles. So both of those factors assure that there's a large diameter in the vascular tissue, so a lot of water can be fitting and flowing through very efficiently. So your concept check for this section. How would a reduction in the number of aquaporin channels affect a plant's ability to adjust to new osmotic conditions? How would long-distance transport of water be affected if tracheids and vessel elements were alive at maturity? What would happen if you put plant cells in pure water? Okay, all of those, you can find the answer from what we just discussed in this video. But as always, if you need help, I am happy to talk to you whenever you need it. I'll see you later. Bye.